Right. Good afternoon, everybody. Thanks, Bobby, for that introduction. And I'm going to use a wireless mic, and hopefully if you guys can hear me, is that sound okay? Perfect. And thanks for Humboldt State University and all the sponsors for the invitation to speak here, because I feel that this information, as well as as you've seen with all the other talks, are very pertinent information, specifically dealing with communities, not just in California, but also nationally. And also, my talk, Is the Grass Really Green? What I'm going to talk about is not, it's not focal to marijuana plants. However, it encompasses it, and it's a variable within this, the conservation issue, and specifically dealing with the conservation perils with the illegal marijuana cultivation in California, and the current knowledge, and so the information, the data that we currently have on this material, but also unanswered questions that we have. The other issue, too, the caveat I'd like to explain is that when I mention about illegal marijuana cultivation, I'm simply talking about trespass marijuana cultivation that's occurring on public, tribal, or community lands. So therefore, these are cultivation that is occurring that is conducted without the public's permission, or the sovereign nation, such as the tribe's permission, or the private landowner's permission as well. So what this talk will cover is current data we have on this issue, the potential impacts to the forest community, so the biodiversity, and barriers in addressing these threats, as well as the potential solutions and future directions. But what this talk will not cover is the ethical or morality issues of marijuana, the medicinal qualities of marijuana, or the political or legality issues. And the reason why is because it's a very emotionally charged subject matter, and it's not me avoiding it, it's the fact that I'm not an expert in this material, and I would leave it better to policymakers, legislators, as well as political scientists to discuss this, because all of these issues specifically can entail a full talk in itself. A little bit of the background on this material is that if you saw Jonathan's talk earlier, talking about in 2004 the listing of the fisher, in 2004 U.S. Fish and Wildlife Service essentially ruled that the fisher is warranted but precluded for listing under the Endangered Species Act. So it's a candidate species. Gives it some protection, but not the full protection that is deemed under that act. And again, going back to the fact that how did the fisher come in wrapping up with marijuana itself, is the fact that we were looking, and one of the main threats to fishers is determining what the causes of mortality is. So that's what we were investigating. We're specifically focusing on life history traits and mortality factors for this species within the state, and that's how marijuana popped up. It's not marijuana itself, but the activities associated with illegal marijuana cultivation popped up as a variable to consider in our analysis. So the next question is, what is a fisher? And it's not a kingfisher. That's something when I'm doing other types of, let's say, it's not visual presentations, and I'm discussing with folks, people always think a fisher is a mammal that's fishing, or a fisher is a kingfisher. But no, it's a mammal which essentially is a forest specialist mesocarnivore or mid-sized carnivore. Its habitat, though, is mid to late stage xeral forest. So what I mean by this is that continuous forest. Fishers the habitat the fishers reside in is not in multifocal fragmented landscapes. They don't prefer edges, meadows, urban, and they're not cosmopolitan species. They're a very rare species, and they prefer mid to late xeral stage forests, and continuous forests specifically. They're also omnivores, so they consume a wide breadth of prey species, and that is encompassing, so they'll eat carrion, so dead animals. They'll consume lots and lots of rodents, and they'll also consume mast, so like acorns, and as well as berries, so manzanita berries, just to kind of give you, they're almost like a bear. Their diet encompasses a lot of the same and overlaps with what a bear would consume. There's a sexual dimorphism, so what I mean by that is that males are slightly larger than females, and the reproduction occurs in March, April, and they'll raise their kits all the way through September. So that's a key point when I bring up the activities that we're going to be discussing about, is that these activities are occurring during the time what is very crucial for population growth, such as reproduction as well as offspring rearing. The other thing is the fishers reside in 2 isolated populations in California. So in California, and that's where I'm focusing this talk is specifically in California. So fishers, there's a southern Sierra population, so it's just south of the Yosemite, the Merced River within the Yosemite National Park, and just south of there to Sequoia Kings Canyon National Forest and Kings Canyon National Park. But that population is very isolated and disjunct from the Northern California population. So the southern Sierra Nevada population is believed to be less than 300 individuals total. So it's a very small population. But the Northern California population, the population estimates are unknown, but it basically resides from the coastal range, so right around here, to the interior coastal range, so right about the I-5 corridor to the Oregon border. There's some reintroduction or translocation efforts that are ongoing, and we wait to see if that population is persistent. But you can see the population essentially is 2 isolated populations, with the Southern Sierra being a very small population. So with this, I'm going to show you a brief video. Again, but if you can see here, this is a key characteristic of what a fisher prefers. This is a decadent old hardwood tree. It's a rest site. And they liken fishers to mini bears, so you can kind of see that. And this is a fisher female coming out of a hardwood den site. So you can see they're preferring these decadent trees with little cavities in there. And you'll also see how agile she is. She's able to shake herself off while she's holding off 50 to 60 feet up in the air. And that beautiful long tail of hers. So now the question is, how did we get to fishers being impacted by marijuana cultivation sites, or the activities associated with them? Like as I mentioned, the causes of death for fishers, specifically within California, or the causes of death for fishers throughout their whole range, was unknown. And it's not well understood. There was anecdotal evidence. We think fishers die of this. We monitored 100 fishers, and we only were able to recover 3 of those, and this is the mortality factors that we assume. And that was most of the data. There wasn't a systematic approach to collecting and determining the causes of mortality. And what I mean by that, there wasn't an approach to start listing them in categories like disease, vehicular strikes, human error. So therefore, since 2006, shortly after the candidate listing for the species, we were able to secure some funding, and we started to look at causes of mortality for this species. And we did this within California, and out of 128 necropsies, we started to determine that. And we started to investigate, again, the main mortality factors. With that, some of the data that we started to generate is we had some surprising results, such as we had a couple individuals die from anticoagulant rodenticide poisoning. And as Jonathan did a great review of what anticoagulant rodenticides are, specifically second generation, which are the super lethal toxic anticoagulant rodenticides out there, we had one fisher die, and essentially what initiated from that, we wanted to investigate that further. So what we did is we looked at what we had archived, liver tissues. And when we started looking at liver tissues for fishers, 80% of these guys were exposed to anticoagulant rodenticides. And these are, as you mentioned in the paper, we looked at public and community lands. And so this is rodenticide poisoning that occurred on fishers that lived in our national park, so Yosemite National Park, our national forest lands, and our tribal community in these sovereign lands. So this isn't fishers being exposed that are residing right in downtown Bakersfield. We're showing that this highly superlethal toxicant is out being dispersed also into the forest, similar to the same rates that people are seeing in other species that are cosmopolitan or within agricultural settings. We also documented 4 mortalities due to this toxicant, so 4 poisonings or toxicosis cases. And then when we looked at the spatial modeling, we tried to determine whether the source points, was this a potential dump site? Someone just went over to the side of the road in the Yosemite National Park and dumped a whole bunch, and that's where we're seeing our exposure rates. But unfortunately, all of our data was showing that it was ubiquitous. It didn't matter where we sampled, the likelihood of us, it was just so ubiquitous in the area we couldn't find essentially a source point for the exposures or the mortalities. And the other very surprising find, data that we were able to illustrate was we had a female fisher, her kit. She died. We went in, we extracted her kit. When we sampled her kit unfortunately died of starvation because she wasn't able to give it milk. And when we sampled that kit was exposed to second-generation rodenticide. What that means is now we have that a kit that is either was exposed to it transplacentally within the womb or was exposed through its mother's milk. And that's a huge finding because now we're starting from not individuals being exposed from consuming prey items. We're talking about individuals being exposed in utero and starting right from ground zero or day one, exposed, and the bioaccumulation can then be compounded onto that. So after the PLOS paper, we started to work with law enforcement, and we wanted to investigate, okay, we have some assumptions, we have some preliminary data that is showing that this toxicant is being used at marijuana cultivation sites. So when we kept on going back and forth with them and gaining access. We were able to see, if you can see in this photo, these little green pellets are actually second-generation anticoagulant rodenticides. So actually, the name brand would be Decon. That's the Decon that was placed all around here, opened up in packages, placed around the plants to protect the plants from rodents that may chew the stalks. So we found massive amounts of this. And we're not talking about, a couple ounces, a pound, 2 pounds. At one site, we were able to document 90 pounds of rodenticide. But it was common for us to find 10, 20 pounds. That was average for us. We also found other toxicants. So in addition to all this is unspent decon. This is packages that we removed from the forest that were out there. So besides having all of this unspent decon, this right here is carbofuran, this is malathion. These sprayers that's pink, which we suspect is carbofuran, which is a banned carbamate that's banned in it's banned in the United States and Canada and the European Union, specifically due to the high toxicity and also the malicious poisonings that occurred due to this toxicant. So we're finding all of these different, it's a huge slurry of toxicants out there. In addition to that we're finding other banned chemicals that are banned in multiple nations, such as DDT. DDT is out in our national forest. We also found fragmented landscapes. This site specifically, just to give you a little bit of a background, has been used for multiple years. So you can see they cleared all the brush, they backed it up into the forest. But this site has been fragmented and has had no ground cover for 6 years to possibly up to 8 years. But this is all the marijuana plants that were growing in these sites. But this is the type of fragmented landscape we're finding, is completely cleared to the ground sites. We're also finding water diversions and toxicant-filled slurry ponds. If you can look at this water, it's slightly blue covered, and the reason why is right around here is probably a couple 100 pounds of soluble fertilizer that folks put in here, as well as other bottles of insecticides. So what they do is they put the insecticides and the fertilizers into these ponds, and then they pipe it out. But this specific pond was is a stream that was completely diverted. Above it, equisetum, other riparian dominated vegetation. Right below it, absolutely everything was dead. So they're diverting full streams that other wildlife species and communities, herpetofauna, aquatic invertebrates, have no more habitat to reside in. So the first paper was able to document the impacts to an individual. We couldn't do a potential population assessment of whether or not this was affecting a specific population, for example, the fishers in the Southern Sierras. This paper that we just published this summer, this paper that we were able to publish this summer was able to document, number one is the average number of marijuana cultivation sites within the female fishers was 5.3 sites. So that means that the fishers that were being actively monitored, and the majority of the fishers in the Southern Sierras were actively monitored and had collars on them, they had an average of 5.3 marijuana sites, cultivation sites within their home range. Now, when we looked at fishers that were exposed to anticoagulant rodenticides versus fishers that were not exposed, the average number was 4 sites for the exposed and then less than 1 site for non-exposed. And the range was actually really kind of eye-popping, where you looked at the exposed fishers had a range of 1 to 16 sites, while unexposed fishers had a range of 0 to 1. So there's a clear correlation that if you had a number of marijuana cultivation sites, you're more likely to be exposed to this specific toxicant. And then we also showed that fishers, that the growth site densities were correlated with fishers' survival rates. We were showing that it had a negative impact on a female fisher survival rate. So therefore now we're able to put into a model or a variable into this model that cultivation sites need to be seriously considered as a factor having some deleterious effect on a fisher's, the population's potential persistence in this small isolated population that has less than 300 individuals. So the new emerging data that we have is that we had additional toxicosis cases, so additional poisonings. We have 3 more, so now based off of the old paper that was published last year was 4. We added these 3, that's a total of 7 new cases. And some of you here who are here locally are familiar with this new case, which is right here of this fisher with the hot dog. So people have labeled it the death by hot dog fisher. And unfortunately what this case is a malicious poisoning of a fisher with a restricted use pesticide. This specific hot dog was laced with methomyl, which is a restricted use carbamate, and it was laced with it, but they had multiple hot dogs around this cultivation site. This fisher died from consuming a hot dog, and the reason why we know this is because the hot dog was positive for methomyl, the fisher was positive for methomyl, and the hot dog that wasn't even digested yet, it was mid-esophagus. That's how acutely toxic this chemical is that the hot dog was mid-esophagus and killed it before that fisher can even finish up swallowing. That's how dangerous that is. These hot dogs, if I would have touched it with a bare hand, the amount of methomyl in these hot dogs would have potentially been a lethal amount for me to touch it. And also the other very interesting item that we found was it was found in less than 20 meters away from marijuana plants in this cultivation site. But the cultivation site wasn't 1,000, 2,000, 3,000 plants. The cultivation site only had 300 plants. So they kind of threw a big, huge wrench into what our analysis is. We thought that major, huge plantations were contributing to the malicious poisons or exposure, but now we have to consider even small plantations, such as 300 plants, or even possibly less, we're also contributing to the impacts that we're seeing. Now, another piece of data is the spotted owl, barred owl data that we're able to generate, is that we've looked at 2 spotted owls, and both of those spotted owls are positive for anticoagulant rodenticide, and specifically the 2nd generation super toxic anticoagulant rodenticide. Now we're looking at barred owls, and when we have 90 barred owl tissue samples that we're testing that have all been collected here locally, and we've only tested 10 of those, and out of those 10, 50% of those are positive. We're working on a publication currently to hopefully get all of this together and mimic very similar to what the Fisher paper is. But again, it's showing another species, such as spotted owls or barred owls, that have the same habitat preferences, such as continuous or mid to late cereal stage forests here in the North Coast or in the Sierras, being exposed to toxicants. Finally, this is actually some very interesting data that we're able to, some pilot data. We're generating additional data pertaining to this, but it's invertebrates. We're able to sample multiple invertebrates at a grow site. And when I talk about this, it's not, okay, here's a huge chemical pile. I'm going to go ahead and sample the dead invertebrates that are around this pile, the mollusk or grasshoppers. What I've done is we've gone to these sites and we'll find a grasshopper or a snail that's in the middle of a grow site, completely alive. There's no lethal, they're not dead basically. And when we tested these, we pulled them up and we tested these samples that were alive. All 5 of them came back positive. And they came back positive, the ones that were alive came back positive for anticoagulant rodenticides, and specifically the 2nd generation anticoagulant rodenticide. The ones that were dead, of course, what we found out was they all were positive for metaldehyde, which is actually the active ingredient in snail and slug bait. So like Corey's snail and slug bait has metaldehyde in there, which is very dangerous. Just to let you know, it's very dangerous if you get a hold of it, or your dog or your kids get a hold of it as well if you inhale it. But all 5 of these came back positive. Now we're talking about invertebrates that there's no deleterious effect for them being exposed to the anticoagulant rodenticide. But as mentioned, the half-life for these are 200, 300 possibly days. That's the half-life. So just to give you a context of that, if it's 1 parts per million, it would take 200 plus days to bring that back down to 0.5 parts per million. And subsequent, it'll biphasically reduce, but again, it can take possibly years for it to fully eliminate. But now we're having insects being exposed to this, so now we're talking at the very bottom level of a trophic food web, prey items being exposed. So a little brief synopsis of what we know is, number 1, we know that a rare forest terrestrial carnivore, such as a fisher, is being exposed to and poisoned by toxicants found in marijuana grow sites. Number 2 is that we know grow sites now with the 2nd publication show that it's impacting fisher survival. And number 3 is spotted owls and barred owls are being exposed. And then finally, we know invertebrates are, the pilot data showing invertebrates are being exposed and they're still alive and potentially can pose a risk to subsequent predators that may consume them. But again, you know, I get a question asked like, well, Mourad, you've only been to so many sites. Is what you're seeing just they're bringing you out to these sites? How extensive is this within the state? Some of you have seen this particular slide previously, but all of these yellow dots are reclaimed grow sites. So these are grow sites that have been fully cleaned up within only 2 of California's 17 national forests. These are just cleaned up grow sites. So again, it would be naive, I think, to say that, oh, it only occurs in these 2 national forests and it doesn't occur up here. But there's many out there, and what I mean by that is Well, the data I'm showing right here, these yellow and red dots are marijuana sites within the Fisher's range for the years of 2010 to 2011. So this is only 2 years of marijuana sites that have been eradicated. And these are trespass marijuana sites. We're not talking your quote-unquote 215. These are trespass marijuana sites. And this is, again, I apologize, it's just most of the work that we've been doing is Fisher-centric. So this slide, we're working on other species, but this slide is Fisher-centric. And what I mean by that is the green polygon represents the current Fisher range, while the light green one represents the historical Fisher range. But there is growth sites all throughout this area. It's just the fact that I'm not displaying that because I'm only focusing on the Fisher. But as you can see here, that and also we need to take in consideration that conservatively, only 40 to 60% of trespass marijuana grow sites are discovered or eradicated. So again, what I'm displaying here is a very, very conservative estimate of what potentially may be out there. And only a fraction of them are clean. And so the sites have the potential, what we're displaying here is the potential to impact 30 to 35% of the Fisher's current range. Now, what I'm going to go through here is basically, again, like I mentioned, I haven't been to that many sites. So one of the questions is, have your team, your collaborators, can extrapolate this to every site that you mentioned in that talk? And the massive use of toxicants, the water diversions, the fragmented landscapes. And I can't state that at the time. I can't state that every one of those over 1,000 grow sites had each one of these variables present. But what I can state is that the 20-plus grow sites that we have been able to visit had all of these present, and some of them in elevated proportions. So let's just kind of focus on what does a specific grow site look like and what the potential impacts to a community may be. So let's just focus on this one portion here in Six Rivers National Forest here in Northern California. One more, there it is. Okay, so this little subset has 30 trespass grow sites within the Trinity River corridor. So out of these 30, let's just go ahead and look at one specific grow site. And there's a grow site here. This grow site actually at one point had close to 80,000 plants. But in the last several years, it only had 15, 20,000. And I believe in the last couple years, it's actually been only about 8,000 plants. But there's a grow site here. There's a massive grow site here. Could you guys see it? You really can't. And the reason why is because I use a pathology term, multifocal. So there's multiple focal points that are dispersed. There's not just this huge cut growing area. It's not this huge area right here. What it is it's right there. All these are watercourses, the blue. And this watercourse actually has the endangered species coho present in there. All these polygons are actually cultivation sites. So let's just go ahead, and I'm going to just focus a little bit on this cultivation site. So, but one of the caveats too I'd like to point out is that, so when people do satellite imagery and they're looking at potential forested canopy, if you were to look at this through some spatial modeling, you would see this as potential forested area. But what it is, right here, is actually a cultivation site. You can see what looks like Christmas trees is actually marijuana plants. This whole site is cleared up, and what they do is they bend brush over so that no one can see it. But this site is down to the rock ground. But you can, it's just, I wanted to kind of illustrate to folks is that it's not very noticeable, and this is Google imagery, and it's really bad, but it's not as noticeable that you're going to see this huge clear cuts that you may see in a logging practice or in what some people are demonstrating in the 215 cultivation sites. Now, The next series of slides are some of the theories or hypotheses that we have, and we're extrapolating them specifically from plausible scenarios that have been demonstrated in other species, but without the specific variable, because it's a new variable, with specific activities such as marijuana cultivation or the impacts from it. But these are directions, some potential scenarios I wanted to illustrate. Can these growth sites facilitate an increased risk to conservation concerned species? So previous thought was fishers, there is some predation that occurs on fishers, but it usually happens to the weak and vulnerable. And what I mean by that is that fishers are going to die because they're heavily diseased. There's a parasite load that's too much for them to handle. They're getting picked off by their predators. And also the vulnerable would be something that, let's say you got hit by a vehicle strike. You got hit by a car, it's got a broken leg, broken jaw, and that's a vulnerable individual that's going to be predated on. That was a previous thought. Current data has shown, though, in California that fishers, the predation is the #1 mortality factor for this species within this state. The bobcat is the #1 predator. That kind of threw a lot of us out, and a lot of people who have been studying fishers for 25, 35 years. That was, to say that bobcats are the #1 predator for fishers was kind of throwing a wrench into a lot of people's thoughts. And the reason why is fishers on the East Coast have been known and documented to kill lynxes that are bigger felids than bobcats. So why are we now having this elevated risk rate of predation occurring in fishers? What are the potential scenarios that are factoring into this, especially for a species that is smaller than another predator of fishers back in the East Coast that these guys are killing? So one of the thoughts we're thinking of, the trail systems that are coming into these growth sites. Again, you're not going to be able to see this from the satellite, but these are mapped trail systems coming into these growth sites. There's trail systems that go into water sources, away from water sources, between escape routes, between and within polygons of growth sites. So all these trail systems here. And we know the data has shown that bobcats, and even current data that's being demonstrated, that bobcats in California or bobcats in other areas of the range, they use basically edge habitats, edge areas. They use road system and trail systems. They're not going to go into continuous, heavily understoried forest lands. So now we need to think about, these are the trails in that growth site. Here's a well-worn path trail with brush that's been cleared, another trail here, and another trail right through this area. And these are trails that are continuous from a skid road that goes all the way down through the continuous forest to these other little multifocal fragmented landscapes. So can these trail systems facilitate conduits for a predator that may not be naturally interacting or interact in a very minimal way, conduits to where the fisher's preferred habitat is, which is a continuous deep forest. Now, the next question is could prey abundance decline due to these pesticides that are being used? So that's specifically rodenticides, and other pesticides as well. And could this be affecting wildlife species indirectly? So what I mean by this is, can these pesticides reduce reproductive rates resulting in lower growth rates or population decline, or expand home ranges to encompass more prey opportunities that increase the movement, and therefore a fisher will encounter, more likely to encounter a bobcat? So one of the questions is there's a massive amounts of rodenticide placed out there. The marijuana cultivation occurs between the months of March and September, October. That's the key point when fishers are reproducing, and the key point when fishers are very highly dependent on prey items to provide that energetic intake that their offspring need. So can the prey for females decline and as you saw there, whisk away prey items, and therefore limit the resources that potentially, with fishers that are usually have an average of 2 kits per litter, there's not enough energetic input for these 2 kits now, and only one of them is potentially going to survive. Is that a potential scenario? It has been demonstrated, not with rodenticide, but has been just demonstrated previously with limited prey items and reduced reproduction and growth rates. And also, the superior competitors, such as bobcats, that are now out competing for fishers and other carnivores, so the fishers don't find enough prey items because these other competitors are in there and basically competing for the same prey. Now, next question is expanding the home range because of this depletion of prey. But they need to expand their home range because there's not enough prey, and they need to encompass more prey availabilities and opportunities. So is there a heightened competition that leads to this interspecific or integral predation occurring? An example would be, like I placed here, is the bobcats and fishers that compete for prey. And are the bobcats increasing predation rates on fishers? Or what has been shown, has been documenting now, is spotted owls being outcompeted by barred owls. Are these species now potentially having this interaction through the potential plausible scenario that the depletion of prey items because of all those pesticides being used at these sites? So this increased home range, and I'll go a little bit with this slide in more detail. And this is this area that I was mentioning that had 30 growth sites within 6 Rivers National Forest, so the national forest right here in our backyard. The yellow polygons represent male fisher home ranges, while the blue polygons represent female home ranges, and the red represent bobcats. So typically what occurs is males' home ranges do not tend to overlap. They usually are adjacent or butt up to each other, while there will be multiple female home ranges that overlap with each other. So as you can see here, you have 3 male home ranges. They're adjacent and butting up to each other, but each one of these home ranges are encompassing numerous females to increase the likelihood of mating opportunities. And bobcats do not prefer the interior forests and prefer this edge road system. So I put the bobcats here. And this is, again, this is a plausible scenario. This isn't actually, we don't have this data currently, but this is a potential plausible scenario where I place these bobcat polygons where road systems are heavy. There's a heavy edge effect right through these 2 areas. So, and I'm going to only illustrate what a potential increase with only one of these individual groups playing a role into this is that if we had what has been documented in previous studies, that if prey decreases, bobcat home ranges increase between 200 and 500%. So conservatively, if I were to increase those polygons by only 200%, it leads to an increased interaction probability with multiple fishers. So as you saw in the previous slide, just to go back, these bobcats were potentially having this interaction with these 2 fishers. So one fisher here and one fisher here. But again, you increase it, you can see the interaction goes to multiple fishers because now the prey abundance has declined and they have to increase their home ranges in order to have the more prey availability. So we're hoping to illustrate this with further data collection. But this is a potential scenario we may be running into. And again, extrapolating it to other species, that there is no data for this. There's absolutely 0 data that our Sierra Nevada red foxes that are being impacted by these growth sites, can these Sierra Nevada red foxes now be having this potential integral predation occurring on them, as well as Humboldt martins, and bobcats or spotted owls and barred owls, as I mentioned previously. Now the next question is, what about the impacts to aquatic organisms within these watersheds? This slide in itself could be a full talk because it is absolutely unknown. The direct and indirect threats to aquatic organisms just haven't been properly addressed. And what I mean by that, there's just a lack of resources, and we're scratching on the surface here. We're starting to ask these questions, but we haven't gone out there and efficiently been able to collect the data to illustrate this. But we're finding an average of 2,000 pounds of dry soluble fertilizer per site. So those 20 plus sites I've been to, I found a fertilizer of 5,000 pounds or 1,700 pounds, but it turns out to be about an average of 2,000 pounds of high nitrogen fertilizer, and an average of 25 gallons of concentrated fertilizer as well. There's also banned and restricted use chemicals, and these fertilizers are just right in the middle of these water courses. We're not talking 5 feet away, we're talking in empty bottles in the water courses where you have these aquatic organisms. You have also, these are tributaries to endangered species such as coho. Now, the toxicants, like I mentioned, the toxicants and fertilizers are poured directly into these water cisterns and diverted. So here's that bluish colored water coming out. But here's a 55-gallon pickle barrel jar. We open it up. We find this toxicant slurry here. And what it is this toxicant slurry was being pumped through a water line that was feeding plants within a riparian corridor. This unknown substance, as well as a dry fertilizer, was right here, where I'm taking the photo, is actually a creek. So these are just right off to the side of a creek. It's just going to take one significant rain event to pump this right into that watershed. So the next question is, are these activities depleting prey for aquatic food webs? Or is it reducing through the high nitrogen loads, the fertigation that's occurring? Is this reducing oxygen levels through algal blooms that could lead to other detrimental effects that people have seen in other places because of this? You know, I think it's the dead zone coming out of the Mississippi River in the Gulf of Mexico. Are we potentially, we got to put all these scenarios up. But again, I cannot state that what we're seeing at these sites, it can be extrapolated to all 1,000 sites. But what we're seeing is really uniform, wanton use of a lot of these chemicals. So the next question is, can this massive use of insecticides be affecting also insectivores? As I mentioned, the invertebrate issue, is many of these toxicants do not affect invertebrates. So we had grasshoppers, we had flying insects that were positive for anticoagulant rodenticide. There's no detrimental effect. They weren't going to die from it. They're just going to sequester it in their tissue. The half-life is there for days, weeks, months in invertebrates. It's been documented before, but no one's done a study is feeding these invertebrates that are positive to a mammalian species or an avian species and see if consuming this tissue in an invertebrate could pose a threat. It's been documented for other prey species, such as rodents, but it's also plausible. But if you have the toxicant in the tissue, it's sequestered there, it's not being utilized and metabolized, it's going to pose a risk to secondary consumers. So that is, again, we're extrapolating, but it's a very plausible scenario. But again, it's unknown if these effects, and that's just only anticoagulant rodenticides. Is it similar to terrestrial carnivores? Could all of this, the malathion, the insecticides that are being dumped out there, create these potential sink areas where there's limited prey abundance for insectivores such as bats or swifts or swallows? Now, on the next slides, I'm going to be talking about the barriers of collecting the data for this specific issue, which has been extremely difficult. It's dangerous, and it's dangerous because there's this misnomer that marijuana is the cultivation of marijuana is organic. It's done by mom and pops, and it's done by very nice folks. And these trespass marijuana grow sites, and I'm not broad brushing it or putting an umbrella that all of it is bad. I'm saying that the sites that we've been dealing with, the trespass marijuana grow sites that are illegal, what we've ran into is armed growers. The toxicants out there, you see a hot dog strung on a triple hook, you naturally want to remove that hot dog. If I would have touched it with my bare hands, I may not be here right now, or a colleague of mine who almost grabbed one may not have been here right now. We may have had a fatality due to that. It's unknown. There's tuna cans, there's sardine cans that are being that are having banned carbamates poured into it. So there's dangerous situations and scenarios out there, not just to myself, but also law enforcement that's trying to clean up these sites, and also the volunteer crews that are trying to clean up these sites as well. And there's also traps, the malicious traps that are used for wildlife out there. The snares, the booby traps are also being, that could potentially pose a risk to us as well. So that's one barrier. The next is the grants and resources. And we're starting to gain a little bit more traction. We're starting to gain a little bit more awareness that this is not an ethical dilemma. That's one of the first issues we dealt with when we started going for grants is we don't see this as an environmental issue. What we see this as is a morality issue and an ethical issue, and your grant is not, doesn't fit the scope of work that we are looking for. We're looking for more ecologically based research, which took us a while to try to illustrate that. It's starting to happen, but it's been very limited for us. It's also very expensive. And what I mean by this is the barriers to collecting data from these reclamation sites is, again, these are over 600 reclamation sites that are here. When this volunteer team came in and they cleaned up the 637 trespass grows, they found 500 additional grow sites of lesser, equal, or greater impacts. And they couldn't do anything about it. They left everything there. The chemicals, the infrastructure is still there. So again, this is a conservative estimate. Add another 500 sites to this, and that's what you have. But that's what's going on because they had no funding. There's a lack of funding for this. And the lack of funding, and I think a lot of us feel, is just the perception that addressing this needs to be only in the halls of legislators. And we feel that we can't wait till policy changes, if policy were to change. We have to start addressing this now, when removing that threat, and reclamation is a key point of that. Now, finally, solutions to this is we need to generate more science-based information. So what I mean by this is it's not science for the sake of science or more science so that I can publish more. It's the fact that we need to have some mechanism to provide more information to inform agencies, inform managers, and policymakers, because a lot of them are completely unaware of this impact. And having our publications out there and doing talks, myself, multiple of my colleagues out there, has been able to disseminate this information, has been very key and instrumental. You just look at the traction it's gained in the last 2 years since it emerged. It's just simply for the fact of disseminating this information has been key. And also educating the public. Here locally, here regionally, a lot of us are aware of this information, but if you go just east of the Rockies, and I've gone to New York and other states and Virginia and done talks, folks are completely unaware of this. They think, again, it's that misnomer that all marijuana cultivation is organic and there's absolutely no environmental impacts that are occurring from it. And that's the case we're trying to illustrate, is that it is not that case. We can't blanket it as it's all bad or it's all good. And also, again, what I'm saying is creating the mechanism of support, because there's just a lack of it, and we need to document it. And what I mean by document it is that what we don't want to have is 20, 30 years down the road, we're seeing an occurrence happen in our forest that we don't have the pieces of the puzzle to put together. We need to start collecting this data, but at the same point, not just the collection of data, we also need to reclimate and remove this threat from our forest by the reclamation efforts out there. We can't just eradicate, cut the plants, and leave all the infrastructure and the toxicants out there. We need to get rid of these threats out. So I'd like to end with a quote from Rachel Carson's book, Silent Spring, where the public must decide whether it wishes to continue on the present road, and it can do so only when in possession of the facts. So we're right here on this trail system. There is still light at the end of the trail. But again, we need to continue down it. We can't just stay in the darkness. We need to continue to illustrate and illuminate what these potential effects are. So with that, if I have time for some questions, I'd like to thank everybody for coming to this talk. Yes, Jonathan.
So you talked about some of the products that are found on the showing some of them being illegal, unavailable in the U.S. and illegal over the counter. Do you think making some of those products that are available now over the counter, not available over the counter, will make a difference in terms of particular second-generation anticoagulants that the Department of Pest Regulation is doing? Or will those simply just come with Mexico or Nevada? Have any thoughts on that?
So the question is whether the over-the-counter toxicants such as second-generation anticoagulants, rodenticides, or even first generations, if having them restricted and there's some type of accountability to it could potentially minimize that, I do think so. I do think that if there's some type of, what I mean by accountability is some type of documentation that can be traceable. That's one thing that we've seen. We run into these restricted use chemicals. So we run into weevilside or methamil and all these restricted use chemicals, and there's no traceability level to it. It's a restricted use. There's no stamping on it. There's nothing. And also the second generation rodenticides are out there that if there was some type of accountability mechanism that we can trace back, and that might be helpful for law enforcement to go back to those applicators and state that your product that you've applied for and you received and you purchased was found in the national forest or our national parks. Can you please explain that? That would be, I think, extremely beneficial as a deterrent. So yes. Yes?
Do you think the increased nitrogen loads coming out of all this excess fertilizer is contributing to some of the blue-green algae blooms that are killing off salmon pretty regularly?
So the question is whether the nitrogen or the fertilizer that we're seeing out there could potentially exacerbate blue-green algae blooms here locally. That is a possible scenario. I think that hasn't been, I know there's folks that are starting to look at it, but it's a very plausible scenario. If you go back to that one site that I showed you, those 30 grow sites, if you were to say that 50% of them were discovered, that's 60 grow sites in only 2 years in that area. And if you were to go ahead and make the calculations that if each one of those grow sites has 2,000 pounds, you know, that over 100,000 pounds of fertilizer out there, and that's only soluble fertilizer, not the liquid concentration. You know, if that runs off with a significant rain event, like what we just had, but then now it's stagnant and it stays in the estuaries, it is a plausible scenario. I think it needs to be investigated definitely further. Yes, in the back. Yes?
Yeah, the money and greed seems to be the big reason why all this marijuana is grown in the first place. So how does that address, what if it became worthless and unprofitable for the plants to be grown there? There would be no reason, no motivation to even pollute the forest in the beginning. Address the economic greed and the money, the motivation behind all that. How can that plant become worthless so there's no reason to grow it?
I've heard that argument in that discussion point. That's just something that I don't feel qualified. I think that's a great question to ask to a political scientist because I'm not avoiding it. It's just the fact that I think what we're showing right now is it's not specifically the plant itself, and I understand that there's various policy that may force it onto our public lands, but it's not marijuana centric because it could be any plant that's out there. Strawberries or corn that's occurring on these public lands would be illegal anyways. But yeah, that's a great point. I've heard that point before. I just can't comment on that right now. Yes?
So I understand putting out, pellets to kill rats because the rats get, damaged in our
Yep.
What is the purpose of the hot dogs? I know that ecological literacy
Yep.
cartel growers is low.
Well, here's the thing is that the malicious poisoning that we're seeing, and the reason why a lot of these chemicals have been banned is let's just say carbofuran or furadan that we found out there. A lot of these chemicals are banned specifically because of the malicious poisoning that has occurred in other countries. And we're seeing that here. We're seeing exactly the same mechanisms. So they're taking what they've used before in other countries and taking that activity and that culture to poison the wildlife. And what we understand from that previous use is that it's to get rid of animals that could potentially damage plants. So what we think is a potential scenario is the fact that bears, large bears that are coming into a grow site and just wandering and tromping around, smashing their plants, they want to deter that. And what we found in that hot dog would have dropped the bear. So a bear would have consumed that hot dog, it would have dropped right there at that point, and that would deter it. We suspect that's probably the issue. It's not. We really don't suspect that these guys have a vindictive against fishers. They're not trying to target fishers. I think it's most likely for. It's not a deer who's going to eat the hot dog. It's probably going to be a bear. Yes.
First, I wanted to say that, thank goodness, the move to legalize is moving forward, could make a huge difference in terms of these grows. But what I wanted to comment on was I'm still sort of struggling with the business of the fishers and bobcats. Fishers are tough little guys. As you said, back east, they take on lynxes, and they live. And so I'm wondering if, okay, you're saying partly that the bobcats are reducing their prey, but also, if I understood you, you were saying that bobcats are also a direct threat to the fishers. Is that not true?
Yes, bobcats. That's fishers. So the question is whether fishers are bobcats are a direct threat to fishers, and yes. Bobcats do kill fishers. That's the number one predator of fishers in California.
So could that be because the fishers are less healthy, less able to take on a bobcat, given that back east they take on lynxes and win?
That's actually a great question. That's a great question. And so the question is there potential sublethal effects that we're not seeing with these toxicants out there that may be predisposing fishers to be predated on by bobcats? It's a great question. And unfortunately, if you saw Jonathan's talk, it's like the fox watching the hen house. That data is basically needs to be shown, the sublethal effects by the manufacturer, and the sublethal effects are they're not well understood at all for this, but there has been documentation that sublethal effects from these toxicants have been shown to cause 40 to 60% an increase in abortions for anticoagulant rodenticides, and there's also the potential additive effects, so that you get exposed once, you get exposed two. Do you need to hit that threshold of coagulopathy or bleeding disorder to have some lethargy and tiredness, or could you be just right below that threshold and still be tired and weak enough, and therefore hitting that category of being vulnerable to predation? It's unknown, but it's a very plausible scenario.
Has anybody looked at found dead bobcats and been able to see whether they have any?
Oh, anticoagulant rodenticides? We've done necropsy on one bobcat, and we only have one sample, but it's positive, you know. So, but I mean, again, it's not surprising. I mean, if 80% of fishers are being exposed, and right now that data is actually old, the level now is up to 86% of fishers are being exposed. So I would state that if I were to roll the dice, probably we're going to have the same likelihood of having bobcats, which also use the same prey species, being exposed as well. Yes?
Wouldn't this just roughly show that the bobcats are less susceptible than fishers for the same exposure that
Well, that is, so can we be seeing bobcats, or is the data that we're showing, because bobcats are consuming fishers, is the data potentially showing that bobcats are less susceptible to rodenticides? That's the question. And I don't think we can answer that, and the reason why is because we're not actively monitoring the bobcats as we're monitoring extensively the fishers. We have these collars, so we're knowing how they're utilizing the landscape, their factors of mortality from birth to death. The bobcats in these areas are unmonitored. So we don't know if we're having that population turnover or that individual turnover and another individual filling in that home range because that bobcat died of toxicosis or poisoning. It's unknown because it's just not being monitored right now. Yes? That's a great question. And so the question is, how do we know bobcats are killing the fishers? And Dr. Wengert right here is actually, she's the one who's doing the forensic analysis. So we have a bobcat, we have a fisher that's dead, and so what we do is we take it in to do a necropsy, and she's able to swab the puncture wounds and amplify the DNA from those puncture wounds and able to identify it not just to species but also an individual. It's pretty interesting new work. Yes?
Anybody look at snails and slugs as food source for fishers?
Yes, the diet analysis does show, so are snail slugs or other invertebrates potentially food sources for fishers? And yes, that has been documented, they're consuming it. So again, when we were able to show that pilot data, and this is previously before we were thinking more of a rodent mechanism. But when the pilot data, we started getting that invertebrates within the fishers that we're monitoring were showing a positive for anticoagulant rodenticides, that now puts in the scenario that we're talking from the bottom up. But we also need to think about the fact that some of these rodents consume these invertebrates. So the rodents can consume these invertebrates, sequester that toxicant in, never ever see the anticoagulant rodenticide because let's say a flying insect moves from a grow site out to the forest. They can get that exposure. There's no grow site in there, but whatever predator consumes that rodent is most likely to be exposed as well. So we need to think of all these very tangential potential routes of exposure as well.